हेलो हेलो। A very good afternoon to everyone present over here. I would like to welcome you all in session 2 of 3rd International Conference of Integrative Chemistry, Biology and Translation Medicine ICBTM 3.0. The theme of invited lectures is Biology of Infectious Disease and Drug Discovery. Now, I would like to request Professor Dinesh Agrawal, Faculty of Pharmacy, Paher University, to felicitate our respected chairperson, Professor Hemant Kothari, Dean PG Studies, Paher University. Professor Hemant Kothari, PhD, NET, JRF, MCOM, MHRM and FDP IIM Ahmedabad is presently working at Pacific University Udaipur as Dean PG Studies with vast teaching experience of almost 25 years and specialization in subjects related to general management, human resource management, behavioral sciences. He is also BOM member of 2 universities. He has authored 3 books and completed 1 minor research project sponsored by UGC, plus 35 research papers in international and national journal of repute are to his credit, and he is also member of editorial advisory board of 3 international and 1 national journal. Moreover, 20 research scholars have been awarded PhD degree under his supervision, and presently 7 scholars are pursuing PhD research work. Thank you, sir, for being chairperson in this session. Now, I would like to request Professor Dinesh Agrawal to felicitate our esteemed speaker, Professor Mandira Verma Basil from Patel Chest Institute, University of Delhi, Bharat. Now, I request Dr. Preeti Kanwar ma'am to felicitate Professor Mandira Verma madam. Now, I would like to request Dr. Preeti Kanwar to felicitate our esteemed speaker, Professor Vinod Rajendran from Pondicherry University, Bharat. Now, I would like to request Professor Hemant Kothari for chair the session. Thank you.
Thank you very much, Dr. Maya. A very good afternoon to one and all present here. Esteemed guests, speakers, scholars and researchers. It's my proud privilege that I am chairing this session on the session called Biology of Infectious Diseases and Drug Discovery. And to be honest, I am not the subject fellow, but since it's an opportunity, I thought why not to learn something new away from my subject. So I welcome all of you in this post-lunch session of this 3-day international conference today on 8th March 2024. I also humbly request all the participants, they are welcome to have questions, but that too at the end of the session, and so that we can have a smooth conduct of the presentation. So no questions would be entertained in between the presentations. You can have the questions after it, and it would be my grace to address all of them. And with these words, I invite and welcome our keynote speaker of this session, Professor Mandira Verma from Patel Chest Institute, Delhi University, Bharat. She would be delivering her note on unveiling the importance of nanotuberculosis mycobacteria as potential human pathogens. Professor Verma currently serves as Director Professor in the Department of Microbiology at the Vallabhbhai Patel Chest Institute, University of Delhi. Her expertise lies in the early identification of M. tuberculosis, mechanism of drug resistance in M. tuberculosis, and molecular epidemiology of M. tuberculosis. Recognized for her contributions, she has been honored with an overseas associateship from DBT in 2002 and an ICMR International Fellowship in 2012 and membership in WHO SAGE IBD in 2023. With 78 articles and 6 book chapter reviews to her credit, she has received numerous awards and honors. Additionally, she contributes as an editorial board member to various national and international journals. So, without taking much of the time, and we are honored to have her here, so I hand over the session to Professor Mandira. Due to some technical problems, we are just starting with the second speaker, our keynote speaker, Dr. Vinod Rajendran from Pondicherry University, Bharat. He would be delivering his note on zinc pyrithione inhabits blood stage parasites of Plasmodium falciparum in culture. Dr. Vinod holds a position as DST Inspire faculty in the Department of Microbiology. He was honored with Fanny Hesse Teaching Excellence Award in 2022 by the International Multidisciplinary Research Foundation. Previously, he served as National Postdoctoral Fellow at IIT Madras, Chennai, India. Dr. Rajendran's contribution includes over 25 publications and several patents. So, I hand over the session to Dr. Rajendran and welcome him to this session.
Okay, good afternoon all. I'll be discussing about zinc pyrithione as a new anti-malarial. Which can be repurposed for treating malaria. So currently I'm of Inspired faculty in Pondicherry Central University, and so a small introduction on malaria, a little bit. So this disease is clinically very important and one of the top priority list by WHO, and due to the COVID-19 pandemic there is a 5% global increase in the malaria incidence, and there's a high documentation of the malaria associated morbidity, and out of the 5 plasmodium species being reported, falciparum is the most deadliest one, which can lead to cerebral malaria. And the one more problem associated with malaria is that there is a co-infection associated. It could be HIV, mycobacteria and other bacterial infections, and the very important problem what we are addressing is the clinical resistance of the parasite. So in the whole life cycle, my study is only focused on the blood stage parasites. So the question, so what happens when the parasite infects the human, especially I mean, the first initial stage is the liver infection, followed by the blood stage. Blood stage is the most critical symptomatic infection, where most of the vaccine candidates and most of the chemotherapeutic agents are being very well targeted, especially on the blood stage infection. So there is a tremendous search for the new drugs because of the drug resistance to the clinical resistance in P. falciparum. So drug repurposing is one way where people are really looking at. When the SARS-CoV-2 outbreak happened, the first thing immediately came for the treatment is hydroxychloroquine. So hydroxychloroquine is an anti-malarial drug, but that was immediately being repurposed for treating a viral disease. So there was a rationale for implying an old drug, anti-malarial drug, for treating SARS-CoV-2, but it resulted in varied efficacy. So here the logic is that rather than starting with a new drug, synthesizing and working, which is going to take several years to reach trials, the shortest way to come up with a new drug is drug repurposing. So here the approach is that I have chosen zinc pyrithione, which has not been explored and it has not been reported for malarial treatment. However, it has been very well studied in bacterial infection, especially in fungal infection. Most of our cosmetics shampoos do have zinc pyrithione, especially to treat the fungal infection. And keeping this in mind, we have tried to understand whether zinc pyrithione has any antimalarial activity on the blood stage infection. So the current situation really emphasizes the need for new drugs. So everyone starts looking for new drugs, new venture to eradicate malaria. So repurposing is one shortest way, I mean, where existing or old drugs can be used, and this is an alternative strategy to combat any of the infectious disease, where SARS-CoV-2 is one of the greatest example where most of the drugs are repurposed for SARS-CoV-2. So in this present study, we have repurposed zinc pyrithione, which is a widely used antifungal drug for the treatment of malaria for the first time. So by looking at these slides, it's very clear that when we start with developing a new drug and reaching human trials is going to consume around 10 years, whereas when you repurpose a drug, we can almost save 8 years, and in terms of the cost effectiveness and in terms of we need not to do any toxicity studies, so directly we can enter into phase 2 trials and phase 3 and then go for the drug registrations. So the researchers and clinicians are considering this strategy to alleviate the dilemma of the drug shortage, and the major advantage of drug repurposing is where we already have the information of pharmacokinetics, dynamics, toxicity profile, and the drugs have already been established in the original preclinical phase 1 studies. So keeping this in mind, drug repurposing strategy is an alternative to tackle these emerging infectious disease as well as the infectious disease which are highly endemic in nature. So malaria is an endemic disease where most of the populations, the African population and Southeast Asian populations are greatly affected. So coming to zinc pyrithione is a compound having a zinc atom and bonding the membrane permeable ionophore pyrithione, which is an FDA approved drug, broad spectrum antimicrobials, basically for topical applications, but there is no report as such when it is being taken inside. So these two studies which were reported for cancer therapy, where zinc pyrithione was found, when they did a high throughput screening for anti-cancer screening, they found zinc pyrithione had a significant activity in killing the tumor cells by inducing apoptosis, and also this has been validated in mice model where they have given 1 milligram per kg body weight. And the second evidence is that zinc pyrithione also mitigates the human ovarian cancer cells. So these two evidences was strong enough for us to make an attempt to understand and evaluate the antiplasmodial activity of zinc pyrithione on human blood stage parasites in vitro culture condition. So before introducing a drug, these days people are looking, does this drug is a fast acting or a slow acting is a big question. to address this question, we try to understand, does it affect the first generation of the parasites or the second generation life cycle of the parasites? So drugs like chloroquine and artemisinin are basically affect the first generation, and they are classified as fast-acting antimalarials, whereas drugs like doxycycline and lumefantrine are slow-acting antimalarials, which basically affect the second generation cycle of the parasites. So to really understand this phenomena, we did a time inhibition kinetics profile using zinc pyrithion, and these are all the blood stage parasites which are stained with pink, and the unstained ones are the human red blood cells which are non-infected. So the drugs which shows efficacy on these early hours of exposure are classified as fast acting, whereas the drugs which affect the later point of time at 48, 72 and 96 are considered to be slow acting antimalarials. So when we performed a time kinetics growth inhibition assay using a cybergreen 1 assay, which can quantify the DNA content in the parasite, and thereby it gives you a quantification whether the parasite is viable or not. So using the cybergreen 1 assay, we did a time kinetics. We exposed zinc pyrithion at early time points like 12, 24, 48, 72 and 96, and we got a 50% inhibitory concentration was determined. So at the initial point it was 5.8 micromolar, and with time there is a progressive decrease in the IC50 value. As well, the one very interesting point is that this zinc pyrithion is very much effective on both drug sensitive and drug resistant strain, which is Plasmodium falciparum 3D7 and Plasmodium falciparum Indo. So Falciparum Indo is a chloroquine resistant strain, and 3D7 is a sensitive. So if you look at the 50% inhibitory concentration, it is equally effective on both the strains, suggesting that it has an equivalent effect on both sensitive and resistant strains. And as well, we determine the resistance index by calculating the IC50 of PF Indo divided by IC50 of 3D7. So the resistance index value also seems to be very low in nature, suggesting that there is no cross resistance with quinine derived antimalarials. So it's very important that to tackle malaria, it should also be equally effective on resistant strains. So this results clearly suggest that this zinc pyrithion is equally effective on a sensitive as well as a resistant strain, having a lesser selectivity index. So thereby the maximum growth inhibition was observed at the later time point, which indicates that it may have a slow acting potential, because at the 12 and 24 hour there is a 5 micromolar and 1.5, whereas at 96 hour we could see it is 0.6 and 0.7 micromolar range, which is sub-nanomolar in range. Then we try to study whether does it, stage specificity, because it's really very important to understand a drug at which stage is highly susceptible to the exposure of the drug. So we synchronized the culture to ring, and then exposed, then to trophozoite we exposed, and then to Syjon. So looking at the data, so the 50% inhibitory concentration for ring is 2.9, and for trophozoite is 1, and for Syjon is 0.3. So again, when you compare a sensitive and resistant strain, it has equivalent effect on both sensitive and resistant, whereas the mature stage of the parasites, where is depicted here in the image, the mature stage of the parasites are highly susceptible to this treatment. So we reason that what could be the possible effect could be because zinc pyrithion holds zinc in it, so there could be a higher accumulation of zinc replacing the ferric ion, and thereby there is a depletion of ATP, ultimately resulting to parasite death, because the availability of ferric ion is not that much available in the early stage of the parasites, whereas the mature stage of the parasites has more ferric ion, where the zinc gets replaced, and that ultimately results in the death of the parasites. This experiment suggests that it is effective on malarial parasites but more susceptible on to the mature stage of the parasites where there is a significant reduction in the IC50 values. So as per the WHO guidelines, the combination therapy is recommended for malaria. Monotherapy is being completely banned. So thereby whatever drugs is in clinical till now is all combination based approach. Then we further wanted to understand, can zinc pyrithion, if we use zinc pyrithion in combination with standard drugs like chloroquine or dihydroartemisinin, do we get any positive interactions? So when we did this experiment, we did a fixed ratio isobologram method where we have taken 4 different ratios like 4:1, 3:2, 2:3 and 1:4, and we could see there is a positive interaction, but we didn't observe any synergistic effect. Rather, we observed an additive interaction between the zinc pyrithion and the chloroquine, as well zinc pyrithion and dihydroartemisinin, which is evident by the fractional inhibitory concentrations, which is 1.1, 1.1 and 1.3. So thereby it is very clear that if zinc pyrithion is administered in combination, it results in a positive interaction, and where the chloroquine is a fast acting and dihydroartemisinin is also a fast acting drug, and zinc pyrithion is likely to have a slow acting. Thereby having one fast acting and one slow acting could be a new combinatorial approach to tackle the malarial infection. So the zinc pyrithion interferes with multiple targets which has not been studied, but whereas we speculate with other pathogens that these are the possible targets, whereas artemisinin basically affects the ROS generation, depolarization of the mitochondrial membrane, and chloroquine interferes the hemoglobin digestions. So one is a lipid soluble, one is a DHA, and zinc pyrithion is a lipid soluble, and chloroquine is a water soluble, where they have different pharmacokinetics, and this ultimately can reduce the delay in the development of resistance, as well it can enhance the long-term efficacy of the drug. So finally we try to see that, because most of the time the drugs, since parasite survives inside the red blood cells, it should not cause any damage to your host cells, and people sometimes misinterpret that the drugs cause damage to the host cells, and they claim it has an effect on the parasites, antiparasitic. So to really address that question, we exposed this zinc pyrithion to the fresh red blood cells, human RBCs, at very high concentrations, to the therapeutic concentrations, and we could see that at up to 100 micromolar we just observed 1% hemolysis, and thereby even at 300 micromolar we observed only 9.7% hemolysis, suggesting that the antiplasmodial effect is independent of hemolysis. So it only acts on the actively growing parasites. So one evidence to show here is that the currently used artemisinin drug, widely used, is known to cause late hemolysis. Even you give through intravenous, intramuscular, or even oral administration, it is known to be so. Thereby it is very a critical factor to really understand when we come up with a new drug and propose them to have an antimalarial effect, should not have any any damage to your host cells, and thereby we say that there was no hemolysis being observed, and the observed antiplasmodial effect is completely independent of hemolysis. So then we wanted to see the phenotypic after the drug exposure, where there is going to be a blockage in the developmental growth arrest. So we again synchronized the ring, trough and sessions and exposed them at 1.25 micromolar of zinc pyrithion, and then observed them up to 48 hours, the developmental growth progression. We could see that the progression from ring stage to trough is bit arrested, whereas the progression from trough to session and the progression from session to the ring is being arrested, where you could see there is a pycnotic morphology, there is a condensation of the DNA. So these kind of observations clearly states that it is equally effective on all the blood stage of the parasites, but there is a 2 to 3 fold enhancement in the activity when it is being given to the mature stage of the parasites, which is trophozoites and scions. So this particular drug, zinc pyrithion, is very effective on all the blood stages, and this could be one of a promising. So we have not deciphered its mechanism of action. We didn't have done any experiments, but we speculate from the previous literature that what all could be the possible events that can take place, since it has got a zinc in it. So increasing zinc concentration may result in the depletion of ATP, because it has been shown very good activity in bacteria, fungi, where it can damage the iron sulfur cluster. Even the iron sulfur cluster is being conserved in the apicoplast of the plasmodium. So thereby increasing zinc concentration depletes ATP, whereas damaging the iron sulfur clusters can affect the biogenesis of the apicoplast, which is a very unique organelle to the plasmodium. So concluding the study, this is the first study to show it is a promising in vitro blood stage activity, and it has effective on all the life stages of the parasite development, and we did not see any hemolytic effect up to 100 microgram per ml, and combining standard drugs like chloroquine and dihydroartemisinin with zinc pyrithion has resulted in positive interactions, where it may have a tremendous therapeutic potential in against the drug resistance P. falciparum, and further studies are needed to understand its effect in the in vivo condition in animal model. We have not done any studies in animal model, so we are planning to do in Plasmodium burgeri infection. Since zinc pyrithion is a drug which is an antibacterial and antifungal, whenever there is going to be a co-infection like malaria, it can simultaneously target the parasite as well as it can clear the bacterial or any fungal infections. So thereby zinc pyrithion could be one of the promising future drug molecule for malaria treatment and its associated co-infections. So yeah, thank you. Yeah. So if there's any question, I'll be happy to answer.
Although it is approved by FDA.
Yes, sir.
It is for topical fungal infection.
Yes.
It is not approved for oral
But two evidences have shown that for cancer therapy, where they have studied in animal models,
oral cancer, you said.
Oral cancer.
Yeah.
So they have given to animals and studied for cancer therapy.
Yeah.
They could.
The question: anybody has seen in animal toxicity?
Sir.
Anybody has studied?
Yes, this paper has shown there is no toxicity up. They didn't see much toxicity up to 5 milligram per kg body weight.
5 milligram.
Milligram per kg.
All right.
Yes.
What I followed is that only zinc was playing a role in this. Is that so?
But pyrithion is also an antimicrobial.
Yeah,
It may also have some role, but we do not have information
no,
on
then
that.
would it not be a good idea to also check out only zinc and compare it with this? That would
But zinc alone cannot enter the cell. It requires the help of an ionophore to gain them entry access inside the cell.
you could give a zinc ion.
Ma'am?
A zinc ion you could give and
Yes.
then try it out, because it would be a good idea to check it out.
But we have done it, ma'am. Only zinc we have given. It requires 50 micromolar to inhibit the parasites. We have tried that. Only zinc.
And pyrithion in some other form, just
Yes,
to get a clear
sure.
idea that this is not doing the work. It could be.
But-
you have not elucidated.
Yes, ma'am. But the report says pyrithion is also an antimicrobial. But
Agreed.
we don't have the activity of pyrithion alone.
That's what I'm saying.
Yes.
It is a good idea to have a complete study and just giving you a suggestion.
Sure, ma'am.
And you may find an answer there.
Definitely.
Which is that.
Thank you.
All the best.
Any more questions? Okay, then with this we conclude the first keynote speech of Dr. Vinod Rajendran, and I compliment him for making a nice presentation and believe that it's really a time to move forward as India is moving and we want to make it a R&D hub here. So it's good to see people are doing good research work. I compliment him for nice presentation and for good research work. Thank you very much. Thank you. And with this, should I invite you? Okay. Okay. So with these words, I welcome Professor Mandira Verma for his keynote speech, unveiling the importance of non-tuberculosis mycobacteria as potential human pathogens.
Thank you very much, and I'm really sorry for the technical problem in the early stage. So, can you hear me? I just speak. Can you hear me now? I hope you can hear. Okay, so good afternoon, everyone, and thank you, organizers, for having me here. It's always a pleasure to be in Udaipur. It's a beautiful city and steeped in history. So I don't think we can ever have a fill of Udaipur. So I hope I don't take too much time, and you can go for your sightseeing trip. So the topic I'm going to speak about is not as glamorous as the city we are visiting, and that's non-tuberculous mycobacteria. Non-tuberculous mycobacteria are what I call as the benign cousins of Mycobacterium tuberculosis. So just like any other benign and mild forms of things, people don't really speak about them. So it's something that's not really spoken about commonly, but what is important is that they are there. They're there in the environment. They're ubiquitous organisms present everywhere in the environment. So exposure is very common. In fact, we found non-tuberculous mycobacteria in our laboratory tap water. We found NTM in the soil surrounding our institute, even NTM within the in the soil within our institute. So it's that common. But so exposure is common, but people really do not get disease that often, and I'll talk about that a little while later. There's another disclaimer I want to make. I'm not going to talk about any target molecules or drugs. This is a purely diagnostic clinical lecture where I'm basically going to speak about my experience over the past decade in a clinical laboratory and how we've moved from traditional techniques of diagnosis to rapid diagnostics. So how research has really helped us in the field to diagnose the disease in patients. So coming back to NTM, there's no human-to-human transmission. There's no animal-to-human transmission that's known. It's mainly directly from the environment, and with the advent of immunocompromised status of patients, this has become more common than it was earlier. Just a little bit about the history. We've known NTM has been there for a very long time, but it was first recognized in 1868 as tuberculosis in chicken. It was called Mycobacterium avium because of that. It was much later in the 1940s that it was first isolated from a patient, and this patient was a patient of silicosis, and he also had Mycobacterium avium infection. And then, of course, it was people started looking for NTM, and there were more cases, especially in the United States. And now, as I'll show you later, that it's found globally. So NTM, when we talk about them, historically they've been divided into slow growers and rapid growers, depending on whether they grow rapidly within 7 days or they take more than 7 days to grow. So that's how we classify them even now. There are about 200 species of NTM that are known, but very few of NTM are actually pathogenic or are potential pathogens. They're all called potential pathogens because they're not definite pathogens like Mycobacterium tuberculosis. So I've just listed a few of the ones that are commonly found in patients. So the slow growers like Mycobacterium avium, Mycobacterium intracellulare, and this rapid growers like Mycobacterium abscessus, fortuitum, and Kiloni. These are the common ones. Now, since we have 200 species, obviously there's a lot of difference between all the species. And very recently, there was this publication where Gupta et al. Showed, they studied nearly 2,000 core proteins of mycobacteria, and they showed differences between the various species. And they said that instead of just having Mycobacterium genera, we should have one Mycobacterium genus for Mycobacterium tuberculosis and 4 additional genera. So the 4 additional are Mycobacteroides, Mycolicibacter, and so on. So all these have been divided into these 4 genera. Now, the recent reports, if you read them, some of them would include these new names, nomenclature. But we'll continue with the Mycobacterium tuberculosis, with Mycobacterium species. So NTM cause both pulmonary and extrapulmonary infections. The pulmonary disease is more common, and it's caused mostly by Mycobacterium avium and Mycobacterium abscessus complex, or even cancerci. And the other diseases that can occur are lymphadenitis, skin and soft tissue infections, bone infection. Actually, it can cause, any infection can also cause skin infection, but the most common is pulmonary infection. So as I was saying, that with because of the immunocompromised status of patients with the advent of HIV, with people going in for organ transplantation and getting immunosuppressive drugs, the NTM disease has become more common. So immunocompromised patients definitely are more prone to NTM infection. But even immunocompetent individuals, some of them are prone, and the factors that make them prone are these, like elderly, age, more than 65, women more than 50 are more prone to NTM, or there's structural damage, structural lung damage like COPD, that's chronic obstructive pulmonary disease, or bronchiectasis. So these can lead to predispose a person to NTM. Now, why is it that we now started talking about NTM? It's not that they've just emerged. It's also because of a larger aging population. The life expectancy has increased, so people who are predisposed to this disease are increasing in number. Then there are lots of environmental and climatic changes, so the ecological niche of NTM may have changed. There's increased incidence of chronic lung disease. Like, I come from Delhi, which is a very polluted city, and the number of patients with chronic lung disease has increased tremendously, and so has the incidence of NTM. But more importantly is that there's increased awareness among medical personnel about NTM, and also there's widespread availability of technology like CT scan and laboratory infrastructure that can actually look into NTM disease. So just a bit about global epidemiology. We can have like 4 to 14.1 cases per 1 lakh population. But it's basically the graph that I wanted to show, that it's not that every geographical region has the same species everywhere. There are differences. So if we look at the blue regions on the left-hand side, the blue ones are marked for Mycobacterium avium intracellulare, which we see that it's more in Australia. I don't know if the graph is clear, but it's more in Australia. It's seen in North America, but even within the Mycobacterium avium complex, we have Mycobacterium avium and we have intracellulare. So Mycobacterium avium, in fact, is more common in North and South America, whereas Mycobacterium intracellulare is more common in Australia. So there's a vast geographical difference, and that is why we really need to look into these potential pathogens, because the treatment of these organisms is different. So again, recently there was this very interesting paper which showed that there's an increased trend in reporting NTM. Now if you look at the map which is above, that shows the increasing trend of isolation of NTM, and the map which is below shows the disease. So they've differentiated between infection and disease, which means that isolation does not mean disease. One has to look at the clinical significance to call it a disease. So we see that India is red over here in the map above. That means there's isolation, but in the map below it's gray. So there is, people have isolated NTM, but very few people have really connected the dots and seen the clinical prevalence. So looking at this, we tried to, we reviewed some studies from South Asia, and we also included our own prevalence, what we had found in our laboratory. And we saw that most of the studies were from India. So out of the 14 studies that were included in our review, only 7 studies had actually looked for clinical prevalence. The rest had not even looked for clinical prevalence. So this can give a very incorrect connotation, because if we just report it, the patient or the clinician might think that this is the disease, but it's not really causing that disease. So here we can see the box which is in gray is our data. The one on the left hand side shows 65 NTM that were isolated in the lab. The graph on the right hand side, the gray box, shows 16 NTM. So that was the ratio. We got 65 NTM, but only 16 of them were clinically relevant. Then again, we looked into the skin and soft tissue infections. Now, ours is basically a chest diseases hospital, but we do get sometimes we get cases of skin infections and post-surgical abscesses. So we got NTM in those, and most of them were Mycobacterium abscesses, and there were a few which were Mycobacterium fortuitum. But even here globally, we see that the green color is for abscesses and the red is for fortuitum on the left-hand side. So the most common skin and soft tissue NTM is Mycobacterium abscesses worldwide. So why is it that we need to, what are the challenges? Why is it that diagnosing NTM infection is difficult and not like TB? TB, if we find it in a sample, it's definitely there. The patient is started on antituberculosis treatment. But for NTM, we have to look for it a lot, because first of all, it's ignored, because there's so many non-specific symptoms. It's often ignored. There's lack of awareness. And we get a lot of mixed cultures. A lot of times we get Mycobacterium tuberculosis along with non-tuberculous mycobacteria, and we'll pick up MTB, but we ignore the NTM. But that NTM may be responsible for the disease also. And secondly, there's so many species. Sometimes the databases available do not even have those species in them. So so those are the challenges that we face. So we follow the clinical diagnostic criteria of the American Thoracic Society, which says that we should get at least 2 NTM in 2 different samples, or we can get 1 histopathological report and 1 positive culture. And if we get 2 positive cultures, then they should be the same subspecies. Again, I'll discuss our cases a little later, so for it to be clear what I really mean by this. So, and if we do not find 2 cases, but the clinician really suspects, then we have to follow up that patient. So for diagnosis, this is what we do in our lab. We do the Zeal-Nielsen staining. We can do quinine or fluorescent, but Zeal-Nielsen staining, the AFP smear, is the most common that is followed. And if we get an AFP positive sample, but we put up gene expert and that's negative, that gives us an indication that it is NTM. So we follow it further for NTM. And culture is irreplaceable. So for Mycobacterium tuberculosis these days in the clinical labs, we just go in for smear or gene expert. If it's positive, that's it. But for NTM, culture we cannot do without culture. We have to go in for it. So we culture both on solid slants. That's the LJ medium that you can see, the green color, and liquid culture, which is in the midget. That's Mycobacterial growth indicator tube, which has a liquid medium. Once we get culture positivity, then we have to go in for the speciation. So we have to put in a lot of biochemical reactions. So this is where my journey started with the biochemical tests, and this was our condition when we put up the biochemical test. It was very frustrating because we had a battery of tests and they were mostly non-specific. So it was very, very frustrating and very time consuming. And now the right hand side, you can see how research has translated in our field in the lab, that now we have an immunochromatographic strip which we can just load from the midget culture. We load the strip, and if it gives 2 bands, then it's Mycobacterium tuberculosis. If we get 1 band, so it's basically looking for MPT64 antigen. If the 64 antigen is present, it's MTB, and if it's not present, then and we get an AFP positive smear, then it's NTM. So this has really shortened our diagnostic timeline for both MTB and NTM. So disadvantages of the traditional methods, of course, is poor sensitivity, there's low specificity, prolonged incubation. We need people need to be highly trained because we get a lot of contamination in the TB lab because of the long incubation times. So, and we need to follow proper biosafety measures. And that is how the modern techniques like Malditov and HPLC have helped. Now I've put HPLC first because this is still in the research phase. It's not really come into the diagnostic lab because it's so expensive, it's cumbersome. One needs to really extract the mycolic acids. But Malditov is something that can come up in a big way. We have tried Malditov with our samples, but with NTM, the problem is that in Malditov, we need an active growth. So with NTM, it's either slow growers or rapid growers, and to get to the right time point is difficult. But people have worked on Malditov and have got very good results with rapid growers, which is obvious because you just have to wait for 7 days, but not such good results for slow growers. Secondly, even amongst the species, there is a lot of difference. As we can see here, the left-hand side shows the slow growers, the right-hand side is the rapid growers, but within those species, there are some that give better results. Like in the rapid growers, Fortuatum and Perigrinum gives better results in Malditov than in the others. So this has still to catch on. This, the experience with Mycobacteria is still limited. The genotypic assays have really been brought into the clinical labs now. So the genotype Mycobacterium CM-AS is what we use in our labs, and that uses the 23S rRNA gene, and we can differentiate between the commonly found mycobacteria and the rare mycobacteria also. So if we first put up the CM, which means common mycobacteria, and if that is negative, then we put up additional strains, which is AS strip. So this is something that we do every day in our lab. The CM Direct, which is on the right hand side, is a new strip that has come up which can directly be done on the samples. The present strip can just be done on positive cultures. The Direct can be done directly on samples, but it's not available yet in India. Another is Inolipa, which is also used in certain labs, but the genotype Mycobacterium has been endorsed by both National TB Elimination Program and by WHO. So another technique that's usually used, which is easier, I mean not as easy as the line probe assay, but easy to use in labs, is PCR restriction analysis, where there are several targets like HSP65, RPOB that have been used. The first papers is what I've highlighted there. I don't know if you can see it. It's by Terenti et al. They used HSP65 antigen. Another paper which is commonly quoted is by Wong et al. They use HSP65 and they use separate enzymes. They used CFO and SAO. And we used the Wong technique on our samples directly, and we got very good results with AFP positive smears, but not such good results with AFP negative smears. So the problem with this technique was that the band sizes were very tiny, so in some species, and for that we could not run them on agar, so we needed new sieve agarose or metaphor agarose. So we used new sieve agar, which is very expensive. So So we devised a new method where we use the same gene, the HSP65. So these are my students, Garima and Anshika. They've both finished their PhD, of course, a long time back now. But they worked on this, and they used HSP65 gene. But we used, the enzymes that we used were NRU1 and BAMH1. And with NRU1 was a serendipitous finding, and we saw that if both the enzymes are restricting, then it is Mycobacterium tuberculosis, and if either one is restricting, then it's NTM. So we formed an algorithm with capital A, capital B being restriction. If that was there, it was MTBC. Any other combination was NTM. So this was much simpler, and we could finish this entire process in a day if we had the DNA. So we switched to this for our laboratory isolates. The problem was still that we still needed restriction enzymes and all the problems associated with restriction enzymes. So my student Kamal, he worked on another project where we used HSP65 and 1458C gene. And this is again a serendipitous finding because HSP65 is known as a housekeeping gene for MTB, but RB1458C is an efflux gene. We were working on efflux pumps, and we found that RB1458C is very specific to MTBC. So we combined both of them, and if both were amplified, then it is MTBC, that is Mycobacterium tuberculosis complex. And if HSP65 amplified but RB1458C did not, then it was NTM. So again, a very rapid technique, and this is what we follow now because it's so rapid, easily doable, and takes very short time. So glad to say that we're done with biochemicals. We do not put up any more biochemicals, and we identify our strains as NTM or MTB with this way. So with that technique, Chanchal and Kamal, they worked on some 151 isolates of non-tuberculous mycobacteria, and they found them out as NTM by the duplex assay, what we call the assay that I showed previously. So that was a duplex assay, and we found that, and after that we speciated the various species by the CM-AS line probe assay, which I had shown before that. So this entire thing, within a week we could finish a number of isolates. So it's really shortened the time or the technological advances that are taking place. So of course, now the gold standard remains genome sequencing. So for NTM, there are 200 species. A lot of times we do not get absolute results with our duplex PCR. Even if we get a negative duplex PCR, we do not get results with CM or AS strips because they do not have all the species. So we have to go in for genome sequencing. For genome sequencing, the HSP65 is used for slow growers, it's preferred. RPOB is preferred for rapid growers, and 16S23, the ITS region, is also used for both rapid and slow growers. Recently, we found a species where we were facing a lot of problems. So we entered it into several databases, and we did not get any exact answer. So I'll come to that a little later. And now, of course, with whole genome sequencing, things have taken a different turn. So whole genome sequence of our isolates used to cost us, it cost 70,000 a few years back. Of course, we never gave it for whole genome sequencing then, but now WGS is like 8,000 for one sample. So it's come down so much, and so obviously whole genome sequencing can be done. So with this, we were doing conducting a study on normal healthy individuals because NTM, as I said, are environmental isolates. So they're present all over, also present in our oral cavity as transient flora. So when we get transient flora as NTM and we have to diagnose a disease, we do not know whether it's a common transient flora or is it a potential pathogen. So for that, we just wanted to see what are the NTM that can be isolated from the oral cavity of healthy individuals. So we took mouth washes and we got Mycobacterium gordoni, simi, chimera, we got fortuitum, perigrinum, a number of isolates, a number of NTM, but there was one NTM which was not identified. So we put up the line probe assay, common strains and additional strains, that was negative because that NTM was not there. And then we studied the 16S, the ITS region, and it was closest to Mycobacterium arupense, which is in the Mycobacterium terrae group. Then it was closest, but it was still not, it was just 93%, so obviously not totally identifiable. Then we did 16S RNA. Again, it was closest to Virginiense, Arupense, Heracleonense, all in the M-terate group, but not exactly either of them. So at this point, we were really confused, and that's why I talked about the databases, that if we are not looking at the correct database, this is what happens, because we do not get a conclusive answer. We did fast ANI, we did taxonomic check on J species WS. With that, we got M. heracleonense closest. Pub MLST said it's Mycobacterium virginiense. And finally, we did a digital DNA-DNA hybridization, followed that database, and the DNA-DNA hybridization said this is a novel species. So we had to go through all those databases. Even now, we're struggling to really prove that it is a novel species. So we have to send it to 2 different databases to get it really verified. But these are the problems with NTM, because any day we can just get a new species. And this was something that this is completely new. So we named this new species SVM VP21. So the SVM is after the S is the person we got it from, V is the person who worked on it later, and M is me. So it's just a name we thought of giving, and VP is of course Vallabhbhai Patel Chest Institute. So this was the 21st odd isolate that we got, so we call it 21. So that's the name we have been given, and now we're thinking of what name to give it. So we thought of Mycobacterium delhi, but it seems WHO bans using names of places, so we have to find a name. No, we cannot do that. Okay, so this was a normal commensal that was found in a normal individual mouthwash. So we did not expect it to be resistant to any of the antibiotics, but we did find that it was resistant to 3 antibiotics. So that is something that one has to be aware of. It's not something that caused a disease, but it's there in the environment. So with that, this is the last part, which you come to the drug resistance in NTM. NTM are ubiquitous, very benign, but very difficult to treat. We have lost around 10 patients in the last 5 years to NTM disease. So if we look at their comparison in comparison to TB, that's maybe 1% of the TB patients. But the TB patients got cured, the NTM patients could not survive. So that is a problem with their therapy. So they are resistant, they're very difficult to treat. I'll just talk about Mycobacterium abscessus because the last case is about Mycobacterium abscessus. Mycobacterium abscessus we treat with clarithromycin, which is a macrolide, which is one of the most important, or an aminoglycoside. So, and there are other antibiotics also, but these are the important ones. So for macrolide, we have to look at the ERM gene, which is an erythromycin methyltransferase gene. And if there is a gene which is functional, then the organism is resistant. If the gene is deleted or if it's non-functional, then the organism is susceptible. So a C to T mutation at the point 28 of the ERM gene shows that it is susceptible. It makes it non-functional. Now, Mycobacterium abscessus has 3 subspecies. So it has Abscessus abscessus, Abscessus maziliense, and Abscessus bolleti. All 3 species have to be treated differently, so we really need to find what is the susceptibility pattern. And maziliense is constitutively susceptible to abscesses, to macrolides, but Abscessus and bolleti are resistant, except for some serovars. So then again, the way we look at drug susceptibility is by the microbroth dilution method, by the MABA method, and that is what CLSI has also recommended. But we also studied several other reports, and there have people who are using ETES, they're using Kirby-Bauer's method, different disk diffusion methods, or some use MABA. So one problem is the uniformity in the drug susceptibility test. Second, with Mycobacterium abscessus, the problem is that it has ERM. If it's functional, it's called inducible resistance. So Mycobacterium abscessus is a rapid grower. It just shows a result in 3 days. If we see a result in 3 days and we remove our isolate as susceptible, that's completely wrong, because we have to re-incubate it. We should re-incubate it for 14 days to see inducible resistance. And this is what happened in our case, where this is one patient that stayed with us for 4 years nearly, 4 years, and really very energetic, happy person. He came in 2019. He was about 67 years old when he came to us first, and we isolated Mycobacterium abscessus from him, and it was susceptible to the macrolides, so he was given clarithromycin, and he improved, and then he went to his son's place in Australia, and he came back very healthy, very happy. So, but then he started getting breathlessness and cough again. So we again tested him. The second time also we got Mycobacterium abscessus, and this time it was resistant to macrolides. So we thought that he has taken macrolides for so long that it's now developed resistance. So his antibiotics were changed. And then he gave us 2 more samples. The last sample came in 2023, which was culture negative. But 3 samples, 2019, 2020, and 2022, again he came back. He did not come to us in 2021. All 3 samples gave us Mycobacterium abscessus. The drug susceptibility MIC was put up, and he was at first treated with macrolides, and then the antibiotics were changed. Later on, recently, when the NTM-DR strip was introduced, again by Hames, that's a recent introduction. It's not a recent, but in India it's a recent introduction. So we started using that, and we saw that the first isolate that we had got was actually Mycobacterium abscessus maziliense, which is constitutively sensitive to macrolides. And the second 2 isolates were the subspecies abscessus, which is constitutively resistant to macrolides. And that is what we got in the strips. So up till the time that we had got this, we could not do whole genome sequencing on this because we did not get the right contigs. But when we put up this test, we finally realized what had happened to the patient. So that is where actually translational research comes in, because what all we do in the lab is actually there going to the patient directly. So we lost the patient recently, unfortunately, because not because of the disease, but with the disease. He died of other causes, but he still had abscesses. He still had the symptoms. So that is how difficult it is to treat NTM, and that is why I thought I would speak about NTM today, because it's very important to make everyone aware that this is something that, even if it's one patient, it's that one patient who's lost his life. So again, this is the last. The challenges are, as I said, it's difficult to first difficult to recognize, difficult to culture. There's so much of mixture, there's contamination, and then there's so many new species. We have to identify them separately because the treatment is completely different. And so how do we bridge the gap? We have to understand the true prevalence. We have to understand that NTM have an impact. Even if it's minor, it does have an impact, so it has to be understood. Development of biomarkers would be really good. Everyone's looking for TB biomarkers, but NTM biomarkers are equally important, because for TB we have several other tests. For NTM, we're really short of good investigative procedures. And of course, we need local guidelines for NTM. So these are our contributors for all our essays that we showed. And we got most of our NTM samples from Rajan Babu TB Hospital and NITRD and Safdarjung hospitals. So with that, thank you. Thank you. So, any questions? So do we have time for questions?
These NTMs are not sensitive to rifamycin. Abscesses, I think, was, isn't it?
Sir, abscesses was, but rifamycin is not the preferred drug for it.
Rifamycin.
Yeah, rifamycin is not the preferred drug for it. Rifamycin is used to treat cancer sign. So instead, for cancer sign, in fact, we just put up rifamycin. If that is resistant, then we put up the other tests. So the normal anti-tuberculosis drugs are not effective on all NTMs.
Otherwise, during treatment, they could have.
Right. So that is why if usually what happens is when the patient comes and he's treated, he's considered as tubercular, he's started on ATT and the patient does not respond, then everyone thinks it's multi-drug resistant TB. Then he started on multi-drug resistant TB and when he doesn't respond to that, that's when people think of NTM.
Amikacin was given in one of them. It was sensitive to amikacin.
Amikacin is fine, sir. Aminoglycosides and macrolides are good for abscesses. So he was sensitive to amikacin, and the second time the patient was resistant to macrolides. So he responded temporarily, but he would always get back. That's the problem with abscesses; it doesn't go away. So whenever we get a patient with abscesses, we're really scared because we know whatever we do, it is a tough call.
Nice presentation, Dr. Mandira. You told us role of MPT64 in the diagnosis of TB and MAVM.
Yeah.
What was your sample and what was the test you performed? Is it the PCR?
So this
sample?
is an immunochromatographic strip, and it's used along with the midget machine. That's the Mycobacterial Growth Indicator tube, which has a liquid culture that's mostly 7H9, although it's a patented thing, so they don't give us the details. But that's a liquid culture. Once it shows positive in the midget culture, then that extract is taken and put into the tube and we get the result within 15 minutes. So it's an immunochromatographic test.
I just wanted to know actual concentrations, sputum or...
No, this is culture. It cannot be done directly on samples. So that's why I said that for NTM, culture is of utmost importance.
Thank you.
It was very interesting talk, ma'am. It actually cleared the role of NTM and their importance. But a little deviated question: do they have you checked presence of PEPP family genes in them?
They have PEPP. So there are some PEPP which are specific for the Mycobacterium TB complex. Those are the PEPP that I was telling you about the other day. But NTM also have PEPP. Yeah.
Because
Yeah.
most of the time it is...
Yeah, it's actually from the same family. It's the Mycobacteriaceae family. So they have a lot of conserved portions between the two.
Thank you.
So with this we conclude the session here, and I am thankful to Professor Mandira for bringing a topic into this conference which is not touched upon very much. And life is important. It is for each and everyone. It is important. It's not the single patient or two, but you have created an awareness and enlightened people. So I am thankful to you for your nice presentation. And I am also thankful to all the audience sitting here, patiently listening to the speakers, and hand over the sessions to Dr. Maya again.
Now, concluding the session with gratitude to all, thank you for your active participation. Your engagement has enriched our session. Until next day, thank you and have a great day. All delegates warmly invited for sightseeing.